Okay, we're going to do the last lecture now, and we're going to cover sections 210 to 214. So our objectives for this lecture are to discuss the structure and functions of lipids, to discuss the structure and functions of proteins, to discuss the structure and functions of nucleic acids, and then to discuss the structure and functions of high-energy compounds. Lastly, we're going to explain the relationship between chemicals and cells. Let's get started. Lipids contain carbon to hydrogen in a 1:2 ratio. They may contain a variety of other elements. For example, they include fats, oils, and waxes. Most are insoluble in water but can be carried in the blood, and they release long-term energy or are used for long-term energy. Fatty acids are long chains of carbon with a carboxyl group, a COOH at the end. Carboxyl can dissolve in water. The carbon chain or tail is fairly insoluble. There are two types of fatty acids, saturated and unsaturated fatty acids. This is an example of lauric acid, and it shows the basic structure of the fatty acid. And you can see the COOH at one end, or carboxyl group. Here we see the difference between the saturated and the unsaturated. The saturated has single carbon-to-carbon bonds, whereas the unsaturated has at least one double carbon-to-carbon bonds. So in the saturated, we have single covalent bonds, but in the unsaturated, we have double covalent bonds. We use fatty acids attached to glycerol to form triglycerides. That's the most common fat in the body. Here are some examples of triglycerides, and you can see how we use dehydration synthesis to build the triglyceride, or we use hydrolysis to break it down. Steroids are the next fatty acid compound. They're large compounds of four connected rings of carbon. Cholesterol is the most common steroid, and they're necessary for the sex hormones. Here we see an illustration of the cholesterol molecule. The third lipid group are called phospholipids. They contain glycerol, two fatty acids and a phosphate group, and a non-lipid group. The non-lipid group is soluble in water. Fatty acids are fairly insoluble. It's the most abundant lipid component in cell membranes. They're called phospholipids. Here we see the groups of lipids that we just spoke of and their functions in the body. We see the lauric acid, and its primary function is an energy source, and it's absorbed from food or synthesized in cells. We see the monoglycerides, diglycerides, and triglycerides. Again, we see they're an energy source, and they're used for energy storage, insulation, and protection. They're stored in fat deposits and must be broken down. Then we see the next one, cholesterol, and we see it's a structural component of all cell membranes, hormones, and digestive secretions and bile. They have the same carbon ring framework. And then finally, we see phospholipids, and they include lecithin, and they're the structural components of the cell membranes. The next macromolecule are the proteins, the amino acids. There's 7 key functions of proteins in the body. They deal with support, movement, transport, buffering, metabolic regulation, coordination and control, and defense. Proteins are long chains of amino acids. There are 20 different amino acids in the body. Amino acids contain a central carbon atom bound to a hydrogen atom, an amino group, an NH2, and a carboxyl group, a COOH. The side chain of varying lengths, amino acids are connected with peptide bonds. This is the structure of an amino acid. You can see the amino group, central carbon atom, the carboxyl group, And last but not least, the R group, and that's a variable side chain with one or more atoms. Each amino acid consists of these things. Here's a sample of peptide bond formation. Again, as we put the two amino acids together, we use dehydration synthesis, removing the water. However, when we break them apart, we use hydrolysis, literally breaking the water apart and adding the OH and the H to the amino acids. Proteins are a function of the specific amino acid sequence. The R group can interact to form a more complex protein, specifically a globular protein. The shape of the protein determines its function. Proteins can undergo something called denaturation. This is a change in protein structure, basically an unfolding. This can happen with changes in temperature. Either too high of a temperature can denature a protein. pH, if it becomes too acidic or too alkaline, or ionic composition. Now, enzymes are important. We spoke of them earlier in the chapter. They're catalysts in the body. What they do is they speed up chemical reactions. There's reactants referred to as substrates. When a substrate interacts with the enzyme, a product is formed. Enzymes have an active site. This is where the substrates bind to undergo their reactions to form products. This is an illustration of how the enzyme actually works. The substrate binds to the active site of the enzyme. In the second picture, once bound at the active site, the substrate is held together, aiding their interaction. In the third illustration, the substrate binding alters the shape of the enzyme and promotes the change and product formation. In the fourth and final illustration, the product detaches from the enzyme and the process is now repeated. The next organic molecule are the nucleic acids. They're large organic molecules and they're made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They store and process information inside the cells. There are two types: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The structure of the nucleic acids are very similar. A nucleotide is composed of a sugar, a phosphate group, and a nitrogen base. DNA is a double strand. The sugar is deoxyribose sugar, and it has 4 nitrogen bases: adenine, guanine, cytosine, thymine. Adenine pairs with thymine, cytosine with guanine. RNA is a single strand. The sugar in RNA is ribose. It also has 4 nitrogen bases: adenine, guanine, cytosine, and uracil. The pairing rule for RNA is A with U and C with G. Here's a table comparing and contrasting RNA with DNA. DNA is double-stranded, RNA is single-stranded. They have 3 of the same nitrogen bases: adenine, guanine, and cytosine. However, DNA has thymine, RNA has uracil. RNA can move in and out of the nucleus. DNA is located in the nucleolus of the nucleus and where it's stored in the genes. Here it shows you the nucleotide structure and the nitrogen bases. This shows you the double-stranded or double helix of DNA and the pairing rule A with T and C with G. Now we move on to ATP, the high-energy molecule. This is a high-energy compound and it's made by attaching a phosphate group to an organic molecule. The name is adenosine triphosphate and it's the most important, and we call it ATP. ATP is made with adenosine monophosphate and 2 phosphate groups. By attaching a phosphate group to the free ADP within the cells, ATP is generated. Energy is stored by converting ADP to ATP, and the reverse releases the energy. Here we show you the illustration of the ATP molecule. You can see the sugar involved in ATP is ribose, and we see the 3 phosphate groups with the high energy bonds. And this shows you the cycle of converting ATP to ADP, then recharging the ADP and converting it back to ATP. And this cycle is constantly happening in the human body. Now this is an overall image of the organic compounds. We have carbohydrates, and they include polysaccharides, disaccharides, monosaccharides. Then we have the lipids. That includes the triglycerides, fatty acids, and glycerol. The proteins composed of peptides and amino acids. The nucleic acids, and they include RNA and DNA, and they're made of nucleotides. And the high energy compounds, ATP, and the nucleotide and the phosphate group. Last but not least, we go to the last section, and what happens is in living cells, we combine the lipids, proteins, and some carbohydrates, and we form structures that work together for a common purpose. Each cell is a miniature organism with structures built by biochemical building blocks. This just shows you the inorganic compounds that we've covered throughout the chapter and the organic compounds. Together, this is how we make up life as we know it, and specifically human life.